Let's talk for a moment about how proteins are folded and how they get assembled. Now, there's different levels of structure that can happen for proteins. We consider a primary structure, a secondary, a tertiary, and a quaternary structure. So what does each level of structure dictate? Well, as you go from primary to quaternary structure, you're dealing with more complex interactions of the amino acids with themselves and then eventually with other proteins. Okay? So in primary structure, we're looking only at the sequence of amino acids. And this sequence is actually specified by the gene itself. So we're simply looking at the peptide bonds that hold amino acids together when we look at primary sequence. In fact, primary amino acids sequence or primary folding, if you want to think of it that way, is so strong that the only way we're going to get primary structure disrupted is either through an enzymatic reaction or by heating a protein up to very high levels and boiling it in acid. Protein structure is very strong at the primary level. At the secondary protein structural level, now we're talking about folding of amino acids into energetically favorable, stable structures. So depending upon the arrangement of the amino acids in their primary structure, there may be a regular arrangement of amino acids that allow that protein to form an alpha helix, that allow it to form a beta pleated sheet. The protein that comes to mind, of course, is collagen. Collagen can form alpha helices. It can form beta pleated sheets because it has a regular arrangement of amino acids that allow it to configure to this alpha helix or beta pleated sheet conformation. The next level of structure, getting a little bit more ordered, is the tertiary structure. And the tertiary structure is basically the three-dimensional structure of a protein. It's held together by hydrophobic interactions, van der Waals interactions, ionic interactions, even in some cases covalent bonding, like disulfide bridges. That's what holds a protein together in its 3D or tertiary level of structure. Now, incidentally, it is the tertiary level of structure that if disrupted, if you disrupt the tertiary level of structure, you denature a protein. You render it useless. You render it non-functional. I always think of a tertiary structure as like when you take an egg, and of course egg is mostly albumin, at least in the whites of the egg it's albumin. What happens when you boil an egg? It turns into a hard-boiled egg. It turns hard, right? It turns hard. Well, you've permanently disrupted the tertiary structure of that albumin. So you'll never be able to get a hard-boiled egg to go back to being a soft egg anymore because you've disrupted that tertiary structure. Now, quaternary structure. Quaternary structure involves all the same properties as tertiary. There's hydrophobic interactions, there's van der Waals forces, there's covalent bonding perhaps. But the difference between tertiary and quaternary structure is that quaternary structure is what holds subunits of proteins together. So for example, hemoglobin. Hemoglobin is a subunit of 4 globin molecules held together, right? 2 alpha, 2 beta globins held together. Well, that would be an example of quaternary structure. Any protein that has multiple subunits, that's quaternary structure. Now, if you denature a protein, of course you're going to lose the tertiary structure. But more importantly, you're going to lose the tertiary structure, and that's what's going to cause a loss of function in that protein. So if you're given a case and you're asked about one of the levels, it'd be more of an experimental type question, but you're asked about the levels of protein structure, just understand primary is the peptide bonds, secondary is hydrogen bonding that holds alpha helices and beta pleated sheets together, tertiary are interactions that make up the 3D structure of the protein, and quaternary structure is what holds subunits of the protein together.